Evolution over billions of years has created huge diversity in life and profoundly shaped the Earth. Fossil fuels formed from living things over millions of years have been rapidly consumed in the last century, resulting in global warming. The chemical sector is heavily dependent on fossil fuels as a raw material and for energy. Bioproduction is an alternative that seeks to produce things in living organisms, generally using single-celled microorganisms like bacteria and yeast. However, achieving sufficient yields in a cost-effective way, enabling replacement of fossil fuels, is difficult. That is why we try to harness the power of evolution to optimize and innovate in the lab. But instead of requiring millions of years, we aim to accelerate evolution. Evolution, as proposed by Darwin, involves diversification, followed by selection dependent on the environment. This occurs over multiple generations. DNA encodes the biological instructions required to produce living things, but is passed down between generations. Changes in the DNA, called mutations, result in diversity being generated and differences between individuals. Mutation of DNA generally occurs at a slow rate over many generations and is broadly speaking untargeted. In the lab, we seek to use and develop the latest tools to enable us to generate high levels of mutation just where we need it in the DNA, in order to achieve faster evolution. Farmers control which animals and plants reproduce over thousands of years to produce the range of crop species and animal breeds we have today. In the lab, we are developing tools to allow us to do the same for microorganisms in rapid automated ways. We hope the tools we develop will help reduce the use of fossil fuels, improve sustainability, and help limit climate change.